involves chewing. The teeth begin the mechanical breakdown of food into smaller pieces that can be swallowed. The salivary glands provide saliva to aid swallowing and passage of the smaller pieces of foods in the esophagus and then on the stomach. The stomach releases gastric juices containing hydrochloric acid and pepsin, which initiate the chemical breakdown of protein. Hydrochloric acid in the stomach begins the protein digestion by denaturing the protein. Denaturation results in the loss of protein's function. Hydrochloric acid also converts inactive pepsinogen to its active form pepsin. Pepsin begins breaking the peptide bonds between amino acids. The powerful mechanical stomach contractions churn the partially digested protein into chyme. The stomach empties the chyme containing the partially digested protein into the small intestine, where majority of the protein digestion occurs. The pancreas secretes digestive juices containing proteases that break down polypeptides into smaller peptides or single amino acids. The two major pancreatic enzymes that digest proteins are trypsin and chymotrypsin. Amino acid dipeptides and tripeptides are absorbed into the cells of the intestinal wall. The cells that line the small intestine release pepsidase that breaks down the diseptides and tripeptides into single amino acids. The muscle contractions of the small intestine mix and propel the amino acids to the absorption sites. In the lower parts of the small intestine, the amino acids are transported from the intestinal lumen to the intestinal cells of the blood, and once the amino acid enters the bloodstream, it distributes them throughout the body.
Understanding amino acid utilization. This study identified the precise net nitrogen utilization, also known here as amino acid utilization (AAU), of various dietary proteins, measures how much of a protein is actually absorbed and used by the body. Also, amino acids produced from the digestions of proteins enter the amino acids pool of the body. So let's define what is amino acid pool. Amino acid pool is the total supply of free amino acids available for use in the human body. Dietary proteins is one of the three sources that contributes amino acid to the amino acid pool. Now, let's understand more about amino acid pool. Number 1. Amino acids from different sources get mixed up to constitute what is known as general amino acid food. Number 2. An adult body contains 100 grams of free amino acid, which is known as amino acid food. Number 3. Amino acid food has no anatomical reality but represent unavailability of amino acid building units. Number 4. It is no functional distinction can be drawn between fate of the amino acid pool synthesized from dietary sources and those derived from the tissue breakdown. And number 5. If a cell takes up as much amino acid as it loses, it is in a state of dynamic equilibrium. And when you say dynamic, it is the change or the progress. And when you say equilibrium, it is the opposing. Opposing forces or influences are balanced. It means dynamic equilibrium is a system in a steady state since forward reactions and backward reactions occur at the same rate. Supplement in dynamic equilibrium, the rate of loss is equal to the rate of gain. Remember that amino acid full if the loss is greater the waste, and if the gain is greater, the cell was gross. Number 6. Another. In order proteins turn over in experimental animal, here are the following orders. First, we have an intestinal muscle, following of kidney, the liver, brain, and muscle. Another explanation of amino acid full. Maybe you are asking on your minds, what is the contribution of amino acid pool in our body? Does it have a contribution? And yes, there is, because amino acids pool contributed by number 1, turnover of body proteins. And when we say protein turnover, it refers to the continual renewal or replacement of protein. It is defined by the balance between protein synthesis and protein degradations. Number 2, its intake of dietary proteins. It is the DRI, or dietary reference intake, is 0.36 grams of proteins per round. Number 3, synthesize is non-essential amino acids. It means it can be synthesized in the body, whereas essential amino acids must be obtained in the diet. Now, let's proceed to the nitrogen balance. So first, what is nitrogen balance? It is a specific terms describe status of nitrogen metabolism in human body. Also, nitrogen balance defined as a balance between amount of nitrogen intake in the form of dietary protein and amount of nitrogen loss unexcreted in the form of urea, uric acid, creatinine, and a small amount of amino acids by an individuals. For more understanding, I will discuss the two types of nitrogen balance, the positive and negative. Negative, so it makes negative when intake is less than loose. It's negative nitrogen balance. Off again less than loose. And if negative, the metabolism in the body may cause some illness or diseases. For example, a chronic or acute illness, protein deficiency, and starvation. Remember that corticosteroid T4 is the hormone that leads to negative nitrogen balance. Next, what is positive nitrogen balance? If positive when intake is greater than loose. Positive nitrogen balance, again, it is greater than. So, positive balance can cause growth hormones, insulin recovery from illness, and the growth hormones. Insulin androgens is the hormones that leads to the positive nitrogen balance.
Transamination reaction is the first step in metabolism of most amino acid. We all know that we have 20 standard amino acids. Out of these 20 standard amino acids, 18 of them can undergo transamination reaction. And in this reaction, the alpha amino group of amino acids are enzymatically removed and transferred to the alpha carbon atom of alpha ketoglutarate, which then become aminated to form L-glutamate, and the reaction is catalyzed by enzyme transaminase or aminotransferases, and the two most important aminotransferases are aspartate aminotransferase and alanine aminotransferase. Transamination is important for distribution of amino groups and production of non-essential amino acid. The amino acid, unlike carbohydrates and fats, it contains nitrogen, and the human body cannot oxidize nitrogen-containing compounds. Before the amino acid can be used as energy source, the nitrogen atoms and their structure must be removed. Then, the carbon skeleton that remains can be processed for energy. We have two stages that obtaining energy through the degradation of amino acid. First is the removal of alpha amino group, and the second one is the degradation of remaining carbon skeleton. The two different types of biochemical reaction are needed for the removal of amino groups from amino acid. They are transamination reaction and oxidative deamination reaction. And now, what is the purpose of amino group? Keto group exchange that occur transamination.
Many different amino acids obtained from protein digestion encountered as reactants and transamination. Keto acids are alpha-ketoglutarate and oxalicetate, with both of which have previously been encountered as intermediates in citric acid cycle. The substance having only two keto acid reactants is the limit of amino acid products to only two, with many amino acid reactants and only two amino acids products. The purpose of transamination do something to one side. Its purpose is to collect the amino groups from many different amino acids into just two amino acids. Therefore, it clarifies greatly the process of amino acid metabolism. In transamination, the amine group on one molecule is exchanged with the ketone group on the other molecule. The amino acid becomes a keto acid and the keto acid becomes an amino acid. Only the two amino acids then need to be processed further with their nitrogen atoms in the end being eliminated from body and urea. And glutamate production via transamination, the most important transamination reaction in protein metabolism. In terms of frequency of occurrence is the one in which the keto acid alpha-ketoglutarate accepts the amino and a lesser extent oxalacetate. Thus, glutamate from alpha-ketoglutarate and aspartate from oxaloacetate are the two amino acids produced in transamination reactions. Further processing of glutamate, there are two pathways for further processing of many glutamate molecules produced via transamination. One pathway is a second transaminase reaction that produces the amino acid aspartate, and the other pathway involves production of ammonium via oxidative deamination. Both the ammonium ion and aspartate, which now become the amino group and carriers that are participants in the urea cycle, and for the aspartate production via transamination in which glutamate is reacting amino acid and oxaloacetate is the reacting keto acid produces aspartate as the new amino acid.
So the oxidative deamination is the process wherein the alpha amino acid is converted into an alpha keto acid with the release of an ammonium ion. This process usually occurs in the liver and in the kidney mitochondria. So glutamine is carried to the liver and the kidney, but mostly in the liver. So going back, glutamine will be broken down and form into a glutamate, and then the ammonium ion is released to the urea. Now, glutamate can now undergo to the process of oxidative deamination. This enzyme can accept or function with either NADP or NAD as a coenzyme. When NAD is used, it will come out as NADH plus H plus, while when the NADP plus is used, it will come out as NADPPH plus H plus. Now, let's go back to the oxidative deamination. I have mentioned a while ago that from glutamate, it will undergo oxidative deamination to form an alpha ketoglutarate and also to release ammonium ions. And since ammonium is a neurotoxic, it will go to the urea and form urine after some process. That's all for the oxidative deamination. Thank you very much for listening.